I'll be presenting on the rose leaf associated metagenomes. What we sampled just for a preliminary study was some select well-established rose plants in the field, and then we sampled progenies from Mother and Fire Glow by Miracle on the Hudson. So altogether, we had about 105 biparental progenies from that biparental cross. And we sampled in a way that we're able to sort of establish a augmented RCBD design. From our sampling, we sampled last year and we're sampling this year also. And each time we take photographs so that we can always go back and rephenotype if we need to. A slide I cut out here was our DNA extraction protocol, where we were doing a leaf wash and then followed through with DNA extraction of the actual leaves to target the endophytes and also use that for genotyping. We're going to update that protocol because there was some miscommunication where we weren't doing the extraction with a buffer to keep the DNA and the RNA stable. So it's sort of underperformed quite a bit. So once we have our DNA samples, what we did was perform the library prep and then sequenced the library. We followed through by demultiplexing and doing quality filtering of the short reads. And then we follow through with metagenomic analysis as well as variant calling. And we do this as a one, so we sequence and we're able to retrieve SNPs as well as metagenomic reads in one single sequencing shot. So we used a software called NGS Composer to do the demultiplexing and quality filtering, and it's a pipeline where you basically upload your data and it does everything for you and spits out the data. For the variance calling, we did we used GVSR, which is also an automated pipeline, and as well as an automated pipeline called QMedi. This is a workflow for the genotyping by sequencing to the bottom right. It's sort of like 4 steps. It's quite streamlined. And we typically do the whole from library prep to sequencing cost us about less than $10 per sample. So this is what the metagenome diversity profile looks like. On this slide, we're not really looking at quantity, relative abundance. We're just looking at how many species we have in the metagenome. So when we do the metagenome analysis at strain level, so we're basically using SNP level resolution for detection of strains. We identified just over 1,000. When we do the profiling at species level, we identify over 2,000 species. And when we do the profile at genus level, we identify 1,200 plus genus. As you can see, most of what we found was bacterial. We do have a significant amount of fungi as well as arthropoda. Like I mentioned, the extraction protocol was very suboptimal for RNA. So I think we didn't even find any virus in this data because I think it basically got degraded during the DNA extraction. But we're going to redo that this year. We're actually doing it right now with an improved DNA extraction protocol. For the relative abundance, it sort of slightly mirrors what you see in the diversity. You still see that bacterial tags are the most abundant, followed by fungal and arthropoda. So looking in more closely, the first thing we wanted to see was, can we actually find verified mites? In this case, we're only looking at the leaf after the wash. So we we're really expecting to see mites in this sample, but luckily enough, we were able to identify mites even after washing the leaf. As you can see if you blow up the arthropoda phylum, you see in this wedge here where you have Phyllocoptis fructifilus. And that was the only species we identified. Now, the database doesn't really contain a reference genome for Phyllocoptis fructifilus, but what we got lucky again because there was ribosomal RNA sequence, ribosomal DNA sequence available in the database, and that's why I was able to pick it up. And of course, there were other species of Phyllocoptis existing in the database that we didn't pick. So we know we know this is probably true. One thing I didn't mention was during the metagenome analysis, we use an approach called an exact matching algorithm. And what that means is a sequence has to match a taxer precisely without matching anything else in the database. So if you have a sequence that matches more than 2 species, we toss it out because it's not diagnostic. So basically, we're only using whatever is left over that's diagnostic for identification and quantification. We also looked at the fungal metagenome, and as you can see here, we have a predominance of Fusarium. This is not unusual for most metagenome profiles we've done. I think Fusarium is quite ubiquitous. And looking more closely, we also want to mention that this field was sprayed with fungicide, I'm sure, right? So I think it was sprayed with fungicide. So we weren't really observing a lot of fungal disease, but regardless, we're still able to pick black spot or the pathogen causing black spot. In this case, we're picking up Diplocarpon rosae specifically, and no other Diplocarpon species. For Botrytis, we're picking up more than 1 species of Botrytis, with the Botrytis Seneca being the second most abundant in the metagenome, as well as Cladosporium. What we did here was to just look at the microbe-microbe interactions. In this case, we're using what we call a CC-LASSO-based correlation network. And what we're doing here is we're saying we know microbes just don't interact in a pairwise way. Like there's a multi-way interaction. One microbe is interacting with multiple microbes. So we sort of control for that background interaction whenever we're looking at each pairwise interaction. And what you can see here is you see what we call microbial hubs in the corallogram. And when you look closer, you see that the Fusarium species tend to cluster together as expected. And we also have this cluster right here that's mostly enriched with species that are known to be sort of have bowel control properties. So they might produce antimicrobial compounds. And here we're also looking at the data, the same data at a higher, including more tags. And you can still, like that's the Fusarium cluster there, and you can see both negative and positive correlations that might indicate antagonism or synergism. And I'll take questions.
Thank you, Vodi.